So in the previous lecture, we walked through a single generation of evolutionary change. This was also cast as a single step out of 1,000. If the difference between phi bar prime and phi bar is on average 1/1000th of a standard deviation, and this is repeated over 1,000 generations, then after the 1,000 generations, the phenotypic average would have been pushed over by one full standard deviation. This would be a big amount of evolutionary change that is accumulated over many generations of microscale evolutionary change. And this is the idea of microevolution. The population in generation T plus 1 was different relative to what it was in generation T. The differences may be very slight, and you probably would never be able to detect the evolutionary change unless you looked at the cumulative change over many generations. But the change is there nonetheless. Natural selection is certainly one potential cause for the population to change from generation to generation, but it isn't the only cause. In this lecture, we'll work through a complete list of all possible causes that could make a population different at generation T plus 1 relative to generation T. There are only 4 really, but I'm throwing in number 5 because it's significant for an important thought exercise that we'll be working through as we continue this lesson into the 4th lecture. Remember, in order to qualify as a microevolutionary force, it has to make the population different from what it was in the previous generation. In order to even begin talking about this, we need to specify how we characterize the population so that we can recognize and quantify the change that occurs. In the previous lecture's example, we used phenotypic average, phi bar, as the thing that changed from generation T to generation T plus 1. We worked through the case where one extreme had the highest fitness. The fitness function is an upward sloping line or downward sloping line. This is called directional selection, and the resulting evolution was a change in phi bar. But we also described 2 other flavors of selection, 2 ways in which you could have a non-random association between fitness and phenotypic value. You could have stabilizing selection, in which an intermediate phenotype has the highest fitness and the extreme phenotypes had lower fitness. And you could also have disruptive selection, in which the extreme phenotypes are the ones with the highest fitness, in which case it might be the average Joe or Jill in the population with the most middle-of-the-road phenotype that has the lowest fitness. Let's look at these in turn. With stabilizing selection, the fitness surface humps in the middle. The phenotype most favored by selection is near the average phenotype. When this is the case, you would expect the successful survivors and reproducers, that is, the parents of the next generation. I drew these in red last time, so I'll do that again here. These will be closer to the mean, and the offspring of those parents should have the same average. No change in phi bar, although they, like their parents, would tend to be closer to the average, having a smaller standard deviation relative to what you would have if there were no selection at all. Here, the change in the population is not in the average phenotype, but rather in the amount of variability, the sigma or standard deviation. Stabilizing selection causes the population to not change in phenotypic average, but be less variable phenotypically than it would be without the selection. I want to remind you that natural selection results in adaptive change. Note that even here, the population in generation T plus 1 has a higher average fitness than the parental generation T because it has more individuals close to the optimum phenotype and fewer with the extreme phenotypes. Selection has caused a tiny step toward a state of greater adaptedness. Disruptive selection is what you have when the average phenotype in the population is also the poorest fitness-wise. In our diagram, this would result in the most extreme individuals having the highest fitness, while the individuals from the center of the phenotypic range would either die or somehow not be well represented in the parentage of the next generation. the expectation should be essentially the opposite of what we saw for stabilizing selection, a greater degree of phenotypic variation relative to what you expect in the absence of selection. I need to mention that there's a little problem with the textbook diagram on disruptive selection. It shows a bimodal distribution in the offspring generation resulting from the disruptive selection. Now, while this could happen, it actually requires a second condition, that the individuals within each of the successful extremes mate preferentially with others in that same extreme. In other words, it requires that mating be non-random. If you have random mating, then the mating pairs would include a majority with one member from each extreme, and these would reestablish a majority of intermediate phenotypes in the offspring. In other words, the distribution should still be unimodal. Broader, that is with a larger standard deviation, but definitely not bimodal. Even with a population at generation t plus 1 being unimodal, the result of disruptive selection is a population that has a higher average fitness than the population at generation t. This is because you have more of the extreme individuals, the more fit ones, in the wider distribution than you have with a narrower one. One thing that is always true of natural selection, be it directional, stabilizing, or disruptive, is that the population ends up changing in a way that results in higher average fitness. Of all the microevolutionary forces, natural selection is the only one that reliably makes the population more fit compared to where it was before the selection. Here's a little thought challenge for you. Given three kinds of natural selection, directional, stabilizing, disruptive, do you think that any of these will be more common or less common in natural populations evolving in the real world? Why? Okay, this was a bit of a curveball, but the line of reasoning one could use here is a great example of how you might line up what we're learning with what you already know. In this case, the way you already understand and envision natural processes. Let's start with a presumption that the organism's habitat is stable over time. The natural conditions that are determining the patterns of natural selection are the same over a period of generations. For any particular trait, natural selection will favor some phenotypic values as good, while others are bad. Let's say the ideal arm length is 30 centimeters. That's where you have the best fitness. And the worst arm length you have is 50 centimeters. We'll show this by plotting fitness on phenotype. This is the top graph in the three-level diagrams we've been using. Values that are close to 30 are pretty good, but they fall off as you move away from the optimum. Phenotypes that are close to 50 centimeters are generally bad, but not as bad as a 50 centimeter arm. So the fitness surface looks something like this, increasing from 25 to 30, then dropping off until you bottom out at 50 centimeters, and then increasing from there. Keep in mind that we're assuming the environment has been pretty stable. So this is the fitness surface that's been in place for at least the past 500 generations. Now, what would happen to a population that were to enter into this world with an average phenotype of 40 centimeters? Right. Correct. It will evolve to the left, but you're right if that's what you said. Its mean phenotype shifts incrementally to the left each generation because the individuals with shorter arms will be more successful at surviving and reproducing, and if arm length is heritable, the next generation will be born with shorter arms on average. Will this evolution towards shorter arms continue forever and ever, with arms getting ever shorter and eventually disappearing entirely? Well, no. You should be able to tell me what's going to happen. Once the average arm length for the population gets to 30 centimeters, there will be no further evolution of mean phenotype. In fact, at that point, the population would be under what kind of selection? Yeah, stabilizing. Now, Would you ever expect this population to evolve towards the phenotype with the worst fitness, here 50 centimeters? Of course not. Natural selection makes a population evolve toward the optimum phenotype, never the worst one. If you look at the fitness surface as an adaptive landscape with peaks where the fitness is highest and valleys where the fitness is lowest, you can extend this metaphor to say that populations must always evolve in the uphill direction. So, if the environment has been relatively stable for a large number of past generations, I would expect populations to be either undergoing directional selection, that is, they're still on their way towards having an average phenotype of peak fitness, or stabilizing selection, because they've already reached the optimum phenotype and are being kept there. Disruptive selection, in which a population's average phenotype is actually the worst of all, should be something that never happens, unless the environment had recently changed, such that under the new conditions the population finds itself in the worst possible position. That's got to be pretty rare, right? In our example, it might have happened if 50 centimeter long arms had been a good thing wherever the population had been before it migrated into this habitat. These are just not common scenarios, and if it's what's required in order for there to be disruptive selection, it must be a rare phenomenon, right? I'm hoping you're seeing how you can use your personal understanding of nature to gain even richer insight from the things we discuss in class. Here, if we can presume that environments tend to be stable, you have to predict that of the three kinds of selection, disruptive selection is the rarest, and therefore least likely to be of great importance in the evolutionary processes that have shaped the biological world. One might be tempted to call this biological intuition, but that would imply that the insight is unfounded by evidence or reason. This position, that disruptive selection is relatively unimportant, is something you could easily defend with sound logic. So far, we've been describing the population before and after natural selection by its phenotypic distribution, the mean phenotype and the standard deviation as a measure of the overall amount of variability. For the remainder of our discussion, we'll turn to the population approach pioneered by Fisher, Haldane, and Wright in their original synthesis of Darwin and Mendel. I'm working under the assumption that you're at least a little bit familiar with the basics of Mendelian genetics, and so terms like homozygous and heterozygous won't cause you any anxiety. If you've never had any genetics before, it's probably no big deal. You can learn what you need from the textbook. Chapter 15, I believe, in Campbell. No need to get too fancy here. In a basic high school type biology class, you will have learned that in a typical Mendelian trait, there are two or more alleles, that is, different forms of a gene, at each genetic locus. We'll follow a single locus at which there are two alleles, big A and little a. With two alleles, there are three genotypes that are possible. You can be big A, or a big A homozygote. You can be little a, or a little a homozygote, or you could be big A, or a heterozygote. If you're good with this terminology, then you should be okay to charge forward in this video without having to brush up in Chapter 15. Now remind yourself here that we're trying to establish a way of quantitatively describing a population. We need this because in order to demonstrate microevolution, there must be a difference between the population at generation T and the same population at generation T plus 1. In population genetics, we basically extend the Mendelian ideas of alleles and genotypes to describe the population by the frequency of the two alleles, big A and little a, and also the frequencies of the three genotypes, big A, little a, and big A, little a. I'll give you an example using snapdragons, in which the two alleles, one for white flowers and one for red flowers, are incompletely dominant, and so the heterozygote has an intermediate phenotype. You have big A being red flowered, little a being white, and big A, the heterozygotes, are pink flowered. Having incomplete dominance is a boon to us with this illustration because it allows us to distinguish easily between all three genotypes, something you can't really do with complete dominance. Going with incomplete as opposed to complete dominance does not affect our conclusions in the least. So let's say you have a population of 100 snapdragon plants, of which 60 are red flowered, 30 are pink, and 10 are white flowered. There are two ways of describing this population. Genotype frequencies. With the calculation of genotype frequencies, you're basically reporting the proportion of the n total individuals that are each of the three genotypes. Here, n is equal to 100, and of those 100, 60 are big A, red flowered. And so the genotype frequency of big A is 60 over 100, or 0.6. We use a capital G for genotype frequency. The genotype frequency of big A, the heterozygote, is 30 over 100, or 0.3. And the genotype frequency of the little a homozygote is 10 over 100, or 0.1. Note that if you add up the genotype frequencies, 0.6 plus 0.3 plus 0.1, you have 1.0. This is always going to be the case for frequencies. This is also the reason why the area under the frequency distribution curve is always normalized to 1. We saw this in the previous lecture. This set of three genotype frequencies is a snapshot in time of our population. From a single snapshot like this, you can't tell if red flowers are on the increase or on the decrease. Some people might want to say, oh, red is most abundant, and so they must be increasing. No, don't do this. You really can't make this conclusion. I know they're the most common right now, but who's to say they weren't even more common last year? If you found out that last year there were 70% or 80% red flowered plants, then the 60% red we're seeing presently is a decrease, right? We don't know how many of any of the genotypes were last year because we're only given data from the present, right now. If you only have a single snapshot in time, you can't tell if selection is favoring red over white or vice versa. In addition to the population's genotype frequencies, you can also describe the population in terms of allele frequencies or gene frequencies. What proportion of all of the genes in the population are big A and what proportion are little a? Now, since we know the genotype frequencies, the calculation of allele frequencies is pretty easy. There are different ways of calculating the allele frequencies. I'll start with the one I like, which is also the quickest. The frequency of the big A allele will be represented by a lowercase p, and it's going to equal the genotype frequency of big A homozygote plus half the genotype frequency of the heterozygote. Here, that's 0.6 plus 1/2 of 0.3, or 0.15, so p is equal to 0.6 plus 0.15, or 0.75. The frequency of the little a allele will be represented by a lowercase q, and it's the genotype frequency of little a plus half of the genotype frequency of the heterozygotes. 0.1 plus 0.15, 0.25. So p is equal to 0.75 and q is equal to 0.25. Like all frequencies, they need to add up to 1, and they do. A second means of calculating the allele frequencies is more labor intensive, but it also makes what we're doing, determining the frequencies of the two alleles in our total population, maybe a little easier to follow. That's what some people say, at least. We have 60 big A, 30 big A, and 10 little a. Now we'll count up the number of copies of the big A allele and the number of copies of the little a allele there are in our 100 individuals. There should be 200 total because each individual carries 2 genes. So our 60 big A will carry 2 copies each of big A and 0 copies of little a. So that makes a total of 120 copies of big A carried by our 60 big A homozygotes. Our 30 heterozygotes will carry 1 copy each of big A and little a. So that's another 30 copies of big A and also 30 copies of little a. Our 10 little a homozygotes will each carry 2 copies of little a, so 20 copies of little a, and of course no copies of big A. Summing up the copies of big A, we have 120 plus 30 or 150, and summing up the copies of little a, we have 30 plus 20 is equal to 50. There are 200 total genes, of which 150 are big A and 50 are little a. P, the frequency of big A, is 0.75, and Q, the frequency of little a, is 0.25. Same result. So in our Snapdragon population, 75% of all the genes are big A and 25% are little a. If natural selection were favoring red flowers over white, you would expect evolutionary change in which P would increase and Q would decrease. Come back in a generation and P should be greater than 0.75, and this would be evolutionary change. If natural selection were favoring white flowers over red, you would expect P to decrease and Q to increase. Any change, delta P and delta Q not equal to 0, constitutes the evolutionary change in our population as it happens over a single generation. Since we only have 2 alleles, it's really not necessary to track both P and Q. They have to add up to 1. So knowing P, you also know that Q is going to be equal to 1 minus P. In our lab exercise this week, we'll be using a simulation program POP-G, which you'll be downloading on your personal computers. Its main graphical output is a plot showing the value of P over time. In lab, we'll have plenty of opportunity to work through a wide array of simulations as needed to illustrate the basic dynamics of our 4 evolutionary forces. What I need to do here, right now in lecture, is to talk about these microevolutionary forces and how they fit into the big picture of evolutionary change. But this graphic, in which P is plotted over time measured in generations, is just a really great way of doing this. I'm not going to spend any more time on natural selection. We'll take our break here and come back to launch into random genetic drift.